drive cargo selectively, and so
Yeah, they tend to do bidirectional transport into the axons.
yes.
And the kinesin, or at least in the paper, they call them kith motors, tend to just deliver into the axon.
Yeah, so I think that one's going to be false too. Okay, number 2. Axons of neurons receive or transmit. That one's going to be
They definitely transmit.
Transmit.
Mm-hmm.
Okay. Information by
Releasing neurotransmitter by
Yes.
synaptic vesicle exocytosis.
Whereas dendrites
Receive.
receive information by neurotransmitters binding to cell signaling.
Yep.
Okay. Axonal microtubules have mixed orientations.
I thought they were no, they're uniform.
Are they uniform? Okay, I was only thinking that because
So
let's see.
If you look at their findings, the axons have fixed orientations, and the dendritic microtubules have mixed.
The mixed because of the plus minus bidirectional thing.
Because the dynesin motors can go bidirectional, meaning
Okay.
that it's not all plus minus in one orientation. Because if you remember, the dynin can only go from positive to negative, and the kinesin goes from negative to positive. But in their testing, at least in the article, they found that only in the dendrites did they see the bidirectional.
Okay, yes, that makes more sense. Thank you. So then the plus ends pointing away from the cell body, was it?
Yes.
I'm gonna check. Okay.
Yeah.
Yeah.
And then dendrites have
Mixed
with the plus
microtubular
ends,
orientations. Mm-hmm.
both and away, both
Yes.
towards and away. Okay.
Yes. I think it would also help to find it in the
Article.
yeah, to find it in the article.
Yeah, it's like right in the background or kind of in the introduction. It's on the first page.
Yes, I found it too.
Where did you guys find? You found it in the introduction?
Background.
Oh, in the background.
Yeah, in the background. So it talks about that. It says
governed
it.
by their mixed microtubule array.
Yes, and then also like right before, like right at the end of the introduction on the first page, kind of.
It says towards the bottom that microtubules and dendrites have uniform polarity opposite to that of axons, and that the minus ends point outwards.
Oh, where are you seeing that?
It's in the intro, just a little bit towards the bottom of the same paragraph.
Same paragraph.
But it doesn't mention anything about the plus ends, just the minus ends.
Oh, here. Whereas in mature neuronal axonal microtubules have uniform orientations with plus ends pointing outward, Microtubules in mature proximal dendrites have mixed orientations. This raises important questions about the establishment of directional transport in dendrites. So its axon microtubules have uniform orientations with plus ends pointing
Away.
outward, yeah, so away from the cell body, which is how the kinesins work. They go from negative to positive, and the dyneins go from positive to negative. Okay. Does everyone feel good about 3?
Did we decide, did we flip the axon and the dendrite then with that?
So
Or
axon
do we keep it
microtubules will have uniform orientations with plus ends pointing away from the cell body.
Okay.
Dendrites have mixed microtubule orientations with plus ends pointing both toward and away because the polarity is mixed.
Sounds good.
The cargoes of microtubule motor transport, protein complexes, and or vesicles can only be transported away from the cell body, down axons or dendrites, but cannot move back towards the cell body. I'm pretty sure that's false.
I think that's false. Yeah. Can only is kind of.
It's actually, I'm pretty sure it shows in their background.
Well, we know for sure that with the dendrites, it's bidirectional and the transport can happen in both directions.
Yeah, that's true. It is bidirectional. Yeah, just the fact that it is bidirectional.
Yeah.
I know they did talk about it though. Well, and some of their tests too, where they altered the proteins, caused gathering in the cell body or in the axon, if you remember.
Yeah. Also, I just found a quote that says in the introduction, motors could move cargo specifically into axons or dendrites. So into meaning like towards the cell body, right?
So
I
yeah,
believe so.
so if it was going from the axon to the dendrite, then you would have to go towards the cell body. But it's whether or not it's being deposited into the cell body. So I don't think I'm able to do it on my phone, but I know on computers, at least, you can search the paper. You can type in keywords such as cell body or soma, and it should help us locate at least where in the paper, because I do know they did talk about that specific phenomenon.
Yes, so right here in the intro cell body, it says, second, the presence of minus end out-oriented microtubules in dendrites suggests that the recruitment of a microtubule minus end directed motor protein dynein could establish selective transport from the cell body to dendrites.
So that still only points to from the cell body to elsewhere.
Okay, also, one of the KIF induced a rapid burst of peroxisomes from the cell body into axon.
So that's still cell body
It's the
away.
same. Okay. We want into the cell body. Peroxisomes move away from the cell body. Let's see.
So we established that it's false, right? Or are you guys looking for answers to make it true?
We're just looking overall
Okay.
to see. Right now it's pointing to where it's kind of going away, but I do know in the article they did talk about aggregates, or I should say vesicles and like peroxisomes aggregating around the cell body, but I'm not sure if it had to do with the deletion or the basically affecting the motor protein so that they couldn't transport away.
Okay.
Okay, one of the results says, these results validate our strategy to use peroxisomes to report on the unidirectional motility of specific motors and show that dyning drives and cargo transport independent of its molecular context. So that like unidirectional thing.
So actually, real quick, the cargoes of microtubule motor transport, protein complexes, and or vesicles can only be transported away from the cell body, down axons or dendrites, but cannot move back towards the cell body. So, for example, if you were transporting cargo, say from the axon to the dendrite, you would have to go towards the cell body, but also the fact that the motor proteins are bidirectional. I think there is near the end of the paper when it's talking about basically movement, that the vesicles are always moving up and down the dendrite to basically provide any receptor components or receptors that are necessary. So by that, they are moving towards the cell body.
Right.
So we have shown that in dendrites of hippocampal neurons where microtubule orientations are mixed, dynin-driven cargo moves bidirectionally. These data indicate that dynin motor proteins do not selectively recognize a subset of uniformly oriented microtubules. Our modeling revealed that bidirectional motility establishes an efficient way to maintain a stable density of particles with a limited number of vesicles, unlike in axons where most cargo needs to be target termini far away from the cell body. Much transport in dendrites eventually targets postsynaptic sites located along the whole length of the dendrite. Quantitative modeling demonstrate that dynin drives cargo from the soma into the dendrites and subsequently maintains a constant density of available cargo by moving it back and forth in the dendrite, meaning it is constantly going towards the end of the dendrite, towards the soma, and kind of back and forth. So I would say that this is actually false.
I would agree with you. That last part, that last sentence, constantly moving back and forth.
Yeah, just kind of really cements that it's kind of moving all around the dendrite, which does mean, yes, it is kind of going back towards the cell body or the soma. Yeah.
And they conclude that it's just a general characteristic of most dendrite vesicles. So I think it's safe to say that it will be false universally.
Yes, because even if the kinesin can't do it in terms of because of the direction, it's not necessarily limited by the soma. It's just where the protein needs to be, or where the cargo needs to be.
Yes.
Match the following microtubule motors with their directionality.
Minus end, right?
Plus end is
dynin is plus to minus. Kinesin is plus and directed.
Oh, I thought dynin was minus and directed.
Me too.
Oh yeah, it is.
So,
in the introduction, it says, sentence, these motor proteins drive cargo transport by moving along microtubules to their plus end, most kinesians or minus end dynin.
Yeah. Because I know dynin is definitely minus n, but kinesian, it's generally plus n, but some of it is minus n. That's why I'm unsure about kinesin a little bit. But just based
see,
on the paper, it says plus N, so we should probably just put plus N.
well, and I know in lecture too, lecture specifically talks about dynein and kinesin as being plus and minus N directed motor proteins. So for dynein, I put minus N directed just because that's where it's heading. And then for kinesin, I put plus N directed.
Cool.
Yeah, because the only reason the dynein can go back and forth is the microtubules aren't a fixed polarization, they're a mix in terms of their polarity.
Right.
The hippocampal nerves used in this study generally have only one axon, but many dendrites. So that would probably be in the introduction.
I think that might be true because of the figures.
Oh, which figure? 1A?
One, yes.
Yeah, it could, but it is a hippocampal neuron, so they can have branching and they can have more than one axon. So I want to see if they mention it in the introduction.
I don't really remember, but I'm looking for it too. They mention it briefly in the intro, but I'm trying to look towards the results for something.
Yeah, see if maybe. So actually, if you look at figure 1A through D, representative images of hippocampal neurons co-transfected at DIV 13 with HA glue R2 and control GFP and stained after 2 days with an antibody to HA red and the axonal marker tau blue. Axonal segments are enlarged to show the HA glue R2 signal in control.
Yeah, if you also look in figure 4 and figure 5, it looks like there's only one axon.
You said
It's
figure
on
4 and
page
figure 5?
6. Yeah, page 6.
Okay. And then you said figure 5 as well.
Yeah, but just in the figure 6 description, it's just saying the hippocampal neuron expressing RFP, visualizing by imaging GFP, the red arrow marks the axon. It doesn't mention axons, like more than one.
Yeah, I would say this one probably wants us to understand like the figures for the most part. This question, I mean.
Yeah, and if you do actually look at figure 7, look at figure 7. It's showing only one axon and it's showing all the different dendrites. So I would go with true. Yeah. Just because if it was otherwise, they would show it in that figure.
Yeah, because if there was otherwise, we'll see the large part twice.
Yeah.
Yeah,
Yeah. Okay.
Okay.
In Capitine et al., the investigators needed to determine which cell extension was an axon. Please click on the fluorescence microscopy data that demonstrates that the cell extension in question is an axon. So I guess we tap one.
I think we got one, yeah.
Okay.
So
Which
So
one?
it's which
which
evidence?
cell extension? Let's go and then compare. Okay.
I'm gonna go back to the paper.
So first is GFP or P50.
So are we just gonna click the first image, the GFP P50, just the middle part?
I think it's the whole image that it wants us to
Yeah, so
determine like which electron or fluorescent microscopy is the axon cell extension,
Oh.
or which cell extension was an axon.
yeah, so
So like which is an axon versus a dendrite? Is that kind of what it wants us to?
yeah, it wants us to select which one is displaying a dendrite and which one is selecting an axon. So please click on the fluorescence microscopy data that demonstrates that the cell extension in question is an axon. I'm assuming that's a typo, but it says in an axon instead
Right.
of is an axon,
Yeah.
which doesn't
Okay,
really.
so the difference between GFP P50 and HA glue R2, it seems like the extension on the glue R2 is like thinner and it has more little like black negative parts. Meanwhile, the P50 is like consistently thick.
So I'm pretty
and
sure
some,
that
yeah.
some of these are staining for the motor proteins and not the actual axon.
Okay.
If we go to the first paragraph of results, it should actually say the answers for us.
Okay.
Okay, so GluR2 will show up in both dendrites and axons because of inhibition they did. So it doesn't exactly point to it being an axon. So I think we can eliminate GluR2.
Yes.
And the GFP P50, that's the whole neuron. That's the whole neuronal cell.
Okay.
I wonder if they're pointing, like they're using the red arrow to indicate like the part of the neuron that we're supposed to be focusing on, or is that just to describe the results, like for the figures?
So I think the red arrow always points to the axon, but I think they want the labeling that was used to determine the axon. So whether it was GFP P50, GluR2, or tau, I think it might actually be tau that was used to determine.
Okay.
But I want to see if I can find it in the writing first before I say anything definitive.
tau is an axonal marker.
Oh, is that what it says? Tau is an axonal marker?
Yes, figure 1, A through D, it says representative images, stained after 2 days with an antibody to HA red and the axonal marker tau in blue. So figures A through D.
So it would be
The
tau
axonal
then.
marker would be tau, right?
Yeah.
GFP is staining for the entire cell body, right?
Yep. Glue is for
Okay.
dendritic transport of, basically it's a dendritic bound protein or receptor. And so by changing dynin, they're seeing how it affects the transport of that molecule.
Yeah.
The GFP P50 is just another stain. And then I think it's just, these are showing the localization of that, whereas tau actually points out what the axon is.
Yeah. Right. I think
Okay.
that's tau. Good. So for that one, there's really only
There's
3
one.
options: GFP, P50, blue, and tau. Correct?
Yeah, and I would say
Mhm.
tau is going to be the better marker.
Yeah. And then did you guys just select like the middle of the image, or did you select the tau word?
I just selected the, like, right by where the arrow is pointing.
Okay, I'll do that too. I feel like
Because they
that
really just
select
want you to
option is
select
a
the
little.
image.
Yeah,
Yeah, but the
yeah.
arrow is pointing to the axon, so.
Okay, sounds good. So
Okay.
next, what are AMPA receptors? What is the GluR2 protein? Are they normally supposed to be in axon or dendrites?
All right,
No.
this should be answered in that
Yes.
first part of the results, I believe.
AMPA glutamate receptors are selectively transported to dendrites in mature hippocampal neurons.
They're dendrite-specific cargo, right?
Yeah, so they are meant to be in the dendrites. If you remember from figure 1, what we were just looking at, and actually, if you scroll down in the results a little bit, it says to examine the role of dynin in dendritic trafficking of AMPA receptor GluR2 subunits, we inhibited dynin function by overexpressing GFP P50 dynetin. We observed that hippocampal dendrite morphology was severely impaired upon dynin inhibition. Moreover, the GluR2 distribution in GFP P50 expressing neurons was dramatically different from controls. GluR2 labeling was no longer exclusively present in dendrites, but now also appeared in the axon. So GluR2 specifically is supposed to just be in the dendrites.
Mm-hmm.
But we need to find out where it talks about the AMPA receptors. If someone does have the search option available, do type in AMPA receptor and see. It might be in the very end, talking about.
Let's see.
Do you guys think it could be AMPA receptor option B? Maybe it's mixed in everyone's, but AMPA receptors are ligand-gated ion channel that detect the neurotransmitter glutamate. GluR2 is one of the protein subunits that form the AMPA receptor complex.
Yeah, at the end, I just saw that it confirms AMPA receptor GluR2 subunit, so that's true with the GluR2 subunit part. I'm still going to look for more AMPA.
Okay, and then like the last part of that answer choice is that since AMPA and GluR2 function to receive, detect neurotransmitter signals, they should normally be in dendrites.
Yes, so it says we know for sure that AMPA is dendrite specific, so that's good.
Mm-hmm. So I think that should be our option right there.
Yeah, I'm just curious where in the article discuss the AMPA receptors because it just kind of starts talking about them, but doesn't give much.
I'm going through, and honestly, I'll read you everything. It's a lot just coming up as hippocampal. So like when I look AMPA, look receptors. Okay.
It's
The
just
first one that they're
describing
talked
them.
about is the intro, yeah.
Yeah, it just describes them as receptor containing vesicles throughout the paper, from what I remember. But that's about it.
So I actually I Googled it too. This is from the National Institute of Health. AMPA receptors are critical for mediating glutamatergic synaptic transmission and plasticity, thus playing a major role in the molecular machinery underlying cellular substrates of memory and learning. So glutamatergic, definitely glutamate that detect the neurotransmitter glutamate. GRG is one of the proteins of the units. I think second option.
Yeah.
What is your second option?
AMPA receptor. It starts with AMPA receptors are ligand gated ion channels and ends with detect neurotransmitters.
Not
they
the axons?
should normally be in the dendrites, not the
axons.
axons. Yeah.
Yeah. Okay. Yeah.
I feel like that's a pretty solid answer choice, but I feel like A is kind of similar, and it also mentions the glumate.
So alpha amino 3 hydroxy 5 methyl 4 isoxazolysyl propionic acid. I think that's just the long name for actually AMPA.
Oh no, not that one. It was like another answer choice. It ends with in both axons and dendrites.
I don't think they would be in both because they're a neurotransmitter receptor.
Okay.
Which you wouldn't really have a neurotransmitter receptor in the axon because it's the site of sending signals versus receiving.
Right.
And GLUA2 or glue, they are subunits.
They are subunits, yes. It says that in the article, so that's good.
So that part's correct.
Yeah, I think this one's good if we want to maybe flag it or come back to it. But I think I think it's good.
You can come back to that.
Okay. So number 9, what is P50 / dynamitin? What is it used for in this paper?
Let's see if it says.
I feel like the first answer is pretty good. The P50 / dynamin is part of a complex proteins that regulate dynamin microtubule motors. Overexpressing P50 inhibits dynamin. It mentions that on page 291.
291.
I remember reading it. It's in the intro, or sorry, not the intro, the results.
Yeah, I'm right there.
Function by GFP tagged P50 dynatin, we observed that hippocampal dendrite morphology was severely impaired upon dynin inhibition. Meaning, so it will impair dynin.
Comparing P50 neurons to GFP controls by measuring the immunofluorescence intensity demonstrated a decrease in the endogenous GLUR2 in the dendrites, as well as an increase in the axon.
I'm not gonna lie, I felt like the organization on this paper could have been better. Like the way they organized all the figures.
Yeah. And they just kept saying whereas and whereas. And I was just like, that's like such a dumb, silly detail, but I was
You noticed it?
peeved by it.
It's a good thing though, because you keep comparing like two major things. And I feel like in our
Yeah.
take-home exam, that's going to be important for us to know.
That's true. That's
I'm
true.
going to be searching the word whereas. Yeah, I feel like answer choice A is pretty solid. I'm not finding anything else about P50.
Okay, which one is your answer choice A?
Yeah, P50, part of a complex of proteins that regulate dynein microtubule motors. Overexpressing P50 inhibits dynein.
Okay. That's my number D, or choice D, so.
For sure, anything that has kinesin in it, it won't be true. But I'm trying to find what is the rule in the paper.
Well, so we know that the P50 dynamitin does inhibit the dynein.
Okay.
Viviane, do you agree that it's the first answer choice? Or
For me, it's number 3.
okay.
Which one is your number 3?
It's the one they're proposing. It's the P50 dynamitin as part of a complex of proteins that regulate dynein microtubule motors.
Yeah.
And then it's overexpressing P50 dynamatin inhibits dynein.
Yes, I agree with that.
Yeah, I will agree. And even just Googling it a little bit, dynamitin is commonly used inhibitor of cytoplasmic dynein-based motility in living cells. Dynamitin does not inhibit dynein directly, but instead acts by causing disassembly of dynactin, a multi-protein complex required for dynein-based movement. So regardless of whether it acts on it directly, it by overexpressing it, you are inhibiting dynein.
Yes, agreed. Okay, number 10. In figure 2, the author uses COS cells, relatively ordinary looking cell line, not nerve cells. They used COS cells to make sure their artificial drug recruitment system was working before they tested it in neurons. In figure 2E to F, based on what you know about microtubule cytophalanges and microfilament, from what is described in the paper and what you know about these motors, approximately where is a centrosome microtubule organizing center? Okay.
The most clear location.
Would that be in the after?
Actually, yes, they do talk about the MTOC in the paper. They do actually bring that up. Actually, if someone could look up the COS cells, because they do talk about those as well. And then once we can find where those are, we will be able to find the discussion on it.
Okay, so figure 2, COS cells are talked about. Let's see where we can discuss it.
I'm seeing it talked about on 296, at least the cost 7 cells.
And then also 294, we first tested in trafficking assay in COS7, which contained radial microtubule arrays consisting of long microtubules running from the centrosome. In the absence of rapalog 1, most proteins were distributed near the cell center and relatively immobile. So figures 2b to 2f might help us.
On figure 4, right?
On figure 4, yes. It says, in absence of rapalog 1, most peroxisomes were near the cell center in figures 2b to 2f in these COS7 cells.
Try typing in mtalk as well. I'd be curious to see if, because I thought I remember them actually discussing that as well.
No, but maybe.
We first tested the trafficking assay in COS7 cells, which contained radical microtubule arrays consisting of long microtubules running from the centrosome. In the absence of rapalog 1, most peroxisomes were distributed nearby the cell center and relatively immobile. And that's saying figures 2b-2f.
So they're injecting, okay, they injected it in a cell line before they tested it, and that separated the microtubules?
So, no, it's... So it's saying without rapalog 1.
Oh, rapalog 1. And then
Which,
without rapalog 1, the peroxisomes. Okay, so what are peroxisomes going to look like? Are they like the really bright white little dots? Are those the peroxisomes?
that's what I'm trying to find out.
I know.
So I know the BICDN inhibits dynin. That was their dynin inhibition complex.
So where would we put our dot? In the before, in figure, I think this is figure 2E, would we put that right in the middle? Because that's where we would put it?
It's asking for the most clear location. So let's go to figure 2E-F and let's read the description for the PEX plus BICDN. So peroxisome distribution before and after rapalog 1 addition in the presence of KIF5 or BICDNF. So I'm assuming they labeled the peroxisomes white.
Yeah, peroxisomes on the outside of that center in the before. In the after, it seems like they all kind of congregate.
So the injection spot should be right in the center, right?
Well, that's where the mTOC will be. That's where all the microtubules come together. I'm going to see where it references it in text. In the absence of rapalog 1, most peroxisomes were distributed nearby the cell center and relatively immobile. Furthermore, rapalog treatment of cells only expressing PEX, RFP, FK, BP did not induce motility. In contrast, addition of rapalong to cells, co-expressing the plus indirected motor proteins KIF5 FRB or KIF17 FRB induced a rapid redistribution of peroxisomes to the cell periphery. Where most microtubule plus ends are located, figure 2b to 2e, consistent with a selective activation of plus N directed motors to selectively probe movement driven by minus N directed dynin. We use the N terminus of the dynin-dynactin interacting protein BICD2 fused to FRB, addition of rapalogs to cells expressing the PEX, RFP, FKB, and BICDN FRB resulted in the strong redistribution of peroxisomes to the center of the cell where most minus N's are located.
Yeah, I have something similar on page 294 where it mentions that they distributed it nearby the cell center as well.
Okay, so let's look up mTOX real quick. So mTOX do actually tend to be near the center of the cell, close to the nucleus, and is the starting point of microtubule growth. So I would argue figure F, the one on the right. It gives us the best location because all those peroxisomes that are tagged are in the center. And that is after the addition of dynin, PEX, and PICDN.
Yeah, that's what I'm also leaning towards.
So I would say it's that.
The brightest white highlight.
Brightest white highlight on the picture on the far right, figure F.
The after.
Yeah. The after.
Yeah.
Yep, the after. Yeah, the after picture for figure F.
Okay, I wanted to explain.
This is going to be a hard one.
Yeah, I think I'll...
When in doubt, go to the conclusion.
So we know for sure that the centrosome microtubule organizing center would be towards the center of the cell or wherever it was injected, which is the center of the cell.
The centrosome is going to be near the center of the cell. So the drug-inducible motor recruitment system works. So it's wanting to answer that one. If you go to the section that says inducible and selective cargo binding of specific microtubule-based proteins. That should have the answer we're looking for.
Okay.
Okay, guys, I think I found it. So if you
Okay.
go to it's going to be 291 to 292. I'm just going to read it off. We hypothesize that activation of specific motor proteins governs the sorting both to axons and to dendrites, and wanted to test whether dynein motor recruitment to cargo is sufficient to establish dendrite-specific targeting. Probing selective motor proteins with endogenous carriers is challenging because these cargoes typically have different types of motors attached. Precluding direct determination of the active type of motor that is driving vesicle motility at any given moment, we therefore developed a well-controlled trafficking assay in hippocampal neurons that allows inducible recruitment of specific motor proteins to drive cargo transport. In this assay, addition of a cell-permeable small molecule during live cell imaging triggers the binding of specific kinesin or dynein motor proteins to stationary peroxisomes, which then report the activity of that particular motor. So it works by using a small cell permeable molecule that triggers the binding of specific kinesin or dynein motor proteins to stationary peroxisomes, and then reports the activity of that particular motor. So how does this drug-inducible motor recruitment system work? Okay, so they use small cell-soluble proteins or molecule that triggers the binding of specific kinesin or dynein motors.
Sorry, I just don't want to type in your guys' ear. That's why I muted.
You're good.
So, sorry, for what I have done so far is that the cell kinesin dynein motors act as a proxy, right? So they can observe how they move through the cell. Is that what you guys are saying?
So
Sorry,
They
my
bind
dog is
to the
barking.
peroxisomes to
that's for the
Yeah.
recruitment of peroxisomes. I think they're more concerned about recruitment of specific motors in terms of increasing or decreasing them.
I think they slow down the.
I think it's
I think it decreases.
okay. So yeah. So actually, if you look at this, these results demonstrate that our strategy successfully recruits the motor of interest to peroxisomes to reduce their motility. So basically, they use a small cell soluble molecule that basically induces the motor protein to recruit peroxisomes, which then induces their motility.
which induces the motility of the motor proteins.
Yeah.
Okay.
So they use a small cell permeable molecule to trigger the binding of motor proteins to stationary peroxisomes. Okay, trigger the binding of motor proteins to stationary peroxisomes, which induces their motility, and by changing factors that affect kinesins, or dinins, they can control which motor is recruited. These factors include these factors include FRB domains and truncated motor proteins. How do you guys feel about that?
Yes, that sounds good. I'm going back to check the factors
So
right now.
I'm on page 294 on the left side of the page.
Okay.
That sounds good.
Yeah.
I think it's important to note towards the bottom, or sorry, on page 295, they end the paragraph by saying these results demonstrate that our strategy successfully recruits the motor of interest to peroxisomes to induce their motility.
Yeah, so I think they also, I think they create their own motors too.
Yeah, because the whole point of this was to report whether or not the motility of the motors were unidirectional or bidirectional. And then they conclude that they are unidirectional.
So I'm going to go, these factors include FRB domains and truncated motor proteins that are able to target specific motor proteins for activation and for imaging fluorescent proteins were added to the complex. There you go. All right. And so just to review everything, I'm going to read my answer off. They use a small cell permeable molecule to trigger the binding of motor proteins to stationary peroxisomes, which induces their motility, and by changing factors that affect kinesins or dyneins, they can control which motor is recruited. These factors include FRB domains and truncated motor proteins that are able to target specific motor proteins for activation. And for imaging, fluorescent proteins were added to the complex. And I'm going to add, and markers added to the peroxisomes.
Awesome.
All right. And I think that's everything, guys.
I think so too. Thank you for recording.
Real quick, I'm going to go ahead and submit, and I can tell y'all how we did.
Okay. What tell you?
It's not telling me how we did. Last time it
Yeah.
did. They probably learned from the last times.
No problem.
All right. All right. I'm going to go ahead and end the
Okay.
recording. I will wait for it to load to the cloud. Did you get a recording as well, Robyn?
It recorded to my laptop because you're the team leader. So I just
Okay.
have like a MP4 file, just in case.
Okay. So worst case, I can message you over Canvas and have you send that over to Garab.
Okay.
Okay. Perfect. All right. I'm going to go ahead and end.
Okay. Thank you so much.
No problem.
Have a great day.
Thanks, you too.